Okay, can you see the screen?
Yes.
It says has the date and name of the class.
Yep.
Okay. All right. So let's have a few announcements. Okay, regarding labs, this week's labs are 17 and 18. So probably you've already downloaded the data for 17. 18 will be available on Wednesday. Now, 17, which started today, involves selective precipitation, and that's similar to one of the review questions in chapter 15. And normally those are some of the more involved sorts of exercises in chapter 15. And FYI, there's a video on Blackboard. It's in the class notes video keys. It goes through one of these step by step in very much detail. And also regarding experiment 18, the post-lab questions are similar to the vitamin C titration lab. And previous labs are due today. That's labs 15 and 16 due tonight by midnight. And by the way, the labs are always due on Mondays by midnight. Okay, another announcement is the third quiz is Wednesday next, April 8th. So that'll be posted on Blackboard by 8 a.m. It'll be due by midnight. And that covers the parts of the chapter 16 class notes that we went over on last Wednesday. That would be pages 11 through 14, covering exercises 10 through 17. So those parts will be covered on the quiz this Wednesday. Okay, and then the class project involving Excel that I've been mentioning off and on since the beginning of class, that'll be due Friday, May 8th, due by midnight. And the details will be gone over tonight, but posted on Blackboard by Wednesday in more detail. And basically it involves using Excel to compare the COVID-19 spike proteins to other spike proteins, like the coronavirus and influenzas activate what's called a spike protein, and that spike protein is what enables it to get inside a cell and inject its DNA. So we'll go over that pretty much tonight. I'll show you the details. Okay, now first for lab 17, are there any questions? So there were a few questions sent by email, like on the procedure and the post-lab questions. Or actually, how about 15 and 16, the ones that which are due tonight, or any lab for that matter? Okay, well, if any question pops up, just let me know. Okay, so this class project is a comparison of the coronavirus spike proteins using Excel. And it involves constructing a graph which shows the differences between the amino acid sequences of the spike protein produced by the COVID-19 virus. And that one has not been officially posted yet. There's literature on it, but as soon as it's official, I'll post that on Blackboard. And then that would be compared to some other spike proteins, such as that of the SARS coronavirus, which is already in the database. Like UniProt's one of the databases where genetic sequences are stored. So that's the first part of this, is making a graph. And then the second part is finding areas where differences occur, and then list the chemical formulas showing how they differ. So this involves aligning the sequences properly and counting the number of differences. This sounds kind of like biology or biochemistry, but it really isn't. Like, those subjects eventually boil down to chemistry if you dig deep enough. So we're going
Mic
to
off.
dig deep and get to the chemistry of it. And then once we finish that, we'll go over chapter 16. There were a couple of examples, and then we'll go on to chapter 17. Okay, does anyone have any questions? Okay. Here I put some information together. It's pretty interesting, and I have some references which I'll post on Blackboard, just FYI. A paper came out from China, it's in February, the beginning of March, and describing what happened. And they said there was a worker at an indoor seafood market where they sold fish and shellfish and also wild animals, including hedgehogs, badgers, snakes, and birds, and various animal carcasses, but no bats. And the reason, of course, they mentioned bats is because the amino acid sequence matches up really closely to a bat, or actually a bat virus. So anyway, this person who was working at this market got sick, and then he had these really weird symptoms, and then they found out he had a different kind of virus. And this particular virus is able to use the human ACE2 receptor to get into the cell, and the human ACE2 receptor happens to be the one that high blood pressure medications target to control or to reduce blood pressure. It's tied into the cardiopulmonary functions of the body. But anyway, so this spike protein, which is generated by COVID-19, attaches to this receptor. And so it was thought that because that's used in medications, that people could be compromised pretty seriously by this virus. And some other bat coronaviruses do bind to ACE2 and some don't. So anyway, these scientists publish this, and it's hard to see because if I'd zoom in a little bit, maybe it would help. So these are the genetic sequences. It's actually amino acid residues. It's their ordering. And the COVID-19's up at the top. All the letters stand for amino acid residues. And for those of you who have taken biology, you're probably familiar with that. So here's a picture of how it gets into a cell. This bottom blue line is a drawing of the cell membrane. And then up here, this top blue line is a drawing of the virus. And the 4 brown things sticking out of the cell are the ACE2 receptors. That's the one that's targeted by high blood pressure medicine. And anyway, so COVID-19 has a gene in it that makes a spike protein, which is this one. It's labeled S1 and S2. They stand for different domains inside that protein, and it attaches to the receptor and basically switches it on. And that tells the cell that it's time to fuse. In other words, the membrane joins with the membrane of the virus, and then the virus injects its DNA inside the cell and uses it as a host to reproduce. Okay, so here's a list of, there are lots of different approaches used to find a solution to this. There are various possible vaccines and therapeutics. There's a list here of all of them. Okay, and then, so let's look at the spike protein. Oh, before I forget, I should turn the recording on, shouldn't I?
Mic on.
I'm also recording the lecture right now. I'm also recording the lecture right now, so I've caught everything up until now.
Okay, thanks.
Mic off.
What are you using to record it?
Mic on.
I'm using OBS Studio. It's a free software for video recording and streaming.
Okay.
Mic off.
Okay, so let's go up here to an example of a spike protein. This is a SARS spike protein. It is 1,255 residues long. And see the, let me zoom in on this. Okay, so here's the end of it. It starts at the first residue. Okay, here, its very first one is M, then F, I, F, and those letters stand for amino acid residues. So they're listed in order from 10 to 50, and then 60 to 100, and so forth. And the or any protein is actually a polymer, and it's composed of a daisy chain bunch of amino acid residues. So they're all connected, and it gets really long and complicated, and they like to curl up into helices. So this gives the impression that they're straight linear molecules, but they're not. They're actually curved. They fold and they have helix formations. And this table here shows the definitions of all the letters. Like the letter R is arginine, H is histidine, K is lysine, D is aspartic acid, E is glutamic acid, and so forth. There are 20 or 21 amino acid residues that make up the proteins, including this one. So the spike protein, for instance, here, let's look for, here's an R. We just looked up R in that table. So if we were actually building a chemical formula for this, we would substitute in for R, one of the models that I just showed you. And the part of the molecule which attaches to the ACE2 receptor is highlighted in yellow here. And this part here at residue number, it's like 716. It's right between the L and the R. That is where the COVID-19 spike protein differs. Like it's pretty much the same until you get right here, and there are 4 new residues inserted there, and that gives it the ability to bind more strongly to ACE2 than previous coronaviruses. So if we were comparing this to the COVID-19, one of the difference would be there'd be roughly 4 new residues inserted right here between the L and the R. So it positions It's like 716 to 717. Okay, so are there any questions?
Mic
Okay,
on.
so somewhere in here I have a link that goes to Uniprot.
Mic off.
But anyway, that's where this was pulled from. So if we went to Uniprot's website and we typed in P59594, this information would be displayed. So this Excel project involves taking these sequences of residues, basically copy pasting this information into something like Word or Notepad or something, and getting rid of all the spaces and making one long continuous chain of letters, and then pasting that into Excel. Okay, so let's look at Excel. Here's a little file I put together just to show basically what's involved. So here is, let's say we're going to just test this, like make it work with, say, 3 residues instead of 1255, just to show the basics. So we're going to have V, R, and A. And I just totally made those up. So here, this column A, here's a position. That's just a number from 1, 2, 3. And I'm not sure how familiar you are with Excel, but it's really by far the most powerful tool that if you're in science or engineering that we have, that comes canned with Windows. So for instance, I typed in 1, 2, 3. If I wanted to not type that in all the time, I could, in cell A26, right here, it has the cell displayed, A26. It's just like the battleship game. If you remember, there's a grid of columns and rows, and you have ships positioned here. Like if a ship was right there, that would be G31. So G31 is displayed there. It's a coordinates. So here, the first position is A25. So in the next cell, we type in plus, which tells Excel a formula is coming. We say A25 plus 1. That'll increment. And then we do a Control C to copy, a Control V to paste. And then, we could copy that all the way down if we wanted to. So now we don't have to type in numbers all the time. So I just entered V, R, and A. And that would come from the information at Uniprot, which originally would look like this. Let's say we have a V and R, or I guess should make them uppercase. Coming straight out of Uniprot, it would look like a row like that, but we need it looking like a column. So we can do a Control C and then go wherever we need to put it, like here, and do a right click. And here where it says paste special, this one right here where it says transpose, will switch rows for columns. See, and it'll paste it as a column instead of a row. And then we can center it using these buttons here to make it look neat. And the other spike residue, which you would choose, it could be anything like a bovine spike protein or mouse human. So you do the same process, look it up on Uniprot, and then paste it down here as a column. So what we want to do is compare these two columns. We want to compare column B to column D. And we can't do that when they're letters. We need to convert each letter to its ASCII code, which is the code that computers use to store letters. Like each letter has a certain number associated with it. A happens to be 65. R is 82. So how we do that is we make a column here and label it code. And in this cell, C25, we ask for it to display the code for cell B25. So that'll convert the V to its number and do the same thing all the way down. So we get 86, 82, 65, and do the same thing for the whichever spike protein you choose. Do the same thing. Now we have these two columns of numbers, and we could subtract them to find differences. If they match, the difference will be 0. So here in column F, we tell Excel to subtract C25 and E25. So take C25, which is 86, minus E25, which is also 86, and the difference is 0. So this is a simple way to compare those two sequences. And we copy paste the formula down, and then here, if we're comparing A to W, of course, there's a big difference, and that's minus 22. Now, the next column involves setting the difference to 1. The reason we want to set it to 1 is so that we don't have points all over the place on this graph. And I had a graph on here, I'm not sure. Oh, there it is. Yeah, here's an example graph. So the first data point is here. Notice when you click on the points, it tells you where the numbers come from. See, it actually highlights them. It says that the X values for this graph come from A25 through A27, which are down here in the positions. So that's where these numbers on the X-axis come from. We have 1, 2, 3 here, and 1, 2, 3 there on the graph. So the Y values come from G25 through G27, which here is false, false, 0, and false is equivalent to 0 here in computer lingo. So we have a 0, and a 1. So anytime there's a difference, we want a 1. You don't want like a 22 or a minus 18 or something. We want to set, anytime there's a difference, we want it to equal 1. And the way we do that is to use the if formula. Excel has tons of formulas. If you go up here to the formulas bar and click around, you'll see all kinds of stuff. Like here's if, and, or, not, XOR, and all kinds of stuff, math and trig and summations, etc. So anyway, for this one, it's the formula is if, and then parentheses F25 less than greater 0, which means if F25 is not equal to 0, then put a 1 in that cell. So here, down here where we do have a difference, the difference is minus 22. So it looks at, here the formula is looking at F27. See, it highlights, if you click on the formula, it shows which which cell is being used. So it's if here F27, which is minus 22, is not 0, which is true, puts a 1 in its own cell. So that's why we have a 1 there. So a 1 ends up displaying here in the graph. It's the third point of the series.
Mic off.
So that's in a nutshell what would be done for the first part. So this, we've compared two sequences, but these are only 3 residues long. You'd be ideally getting this to work with maybe a dozen residues or so and get everything working and get the graph all nice and shiny looking. And then when everything's done and tested and working, then paste in the real McCoy, the 1200 plus residue long sequence, and compare them. So it's important to actually test and make this whenever it's really tiny, like a dozen or so residues. Or it could be even less, like 5 or 6 or even 3. Just make sure everything's working before plugging in the actual residues. So now the second part of this is identifying the differences and counting them. So we've actually already identified the differences. That's what this if statement did. The one value showed that a difference occurred here, and we want to display the residue associated with each protein. So we need the map of the residues, which is here. This tab says amino acid formulas. So if we look at that, it has the chemical formula, what's called the linear structure. If you want to find these on the internet, they're kind of hard to find, but you need to search for linear structure. So for instance, D is aspartic acid, and its linear structure looks like this. You'll see they all have things in common. They have an amino group, and they have an organic acid group, which is COOH, hence their name, amino acid residues. They become a residue when they're put into a protein and attached to one another. So anyway, so we want to look at, for instance, a COVID-19 sequence. If there's an N there, then we want to pull this formula, paste it into the other sheet. And we'd want to paste it here. Or no, not there. We'd want to paste it here. Make a list of the 21 or so amino acid residues, their codes and their formulas, and put them somewhere on the sheet, like right here, or you could use a different sheet if you want. So for example, this one's code A is this formula that corresponds to this here. This is cell A7 and cell E7. So let's do the next one. The next one is R. So how do we paste it over there? Well, we do a copy. When we copy it, it has this moving dashed line around it. We go back over here, click the cell, the target cell, and then right click, say paste special, and tell it to paste a link. There, see, it'll put an R there. Now we need the formula, so we get the formula of R. Copy this, then go back to the main sheet, click where the target is, right click, paste special, paste link. See, now we have the formula for R. And so forth, go down the list and get them all in the sheet. And we want to, in this column here where it says COVID-19, it has an if statement, and this is a little more complicated. So it says if it looks at this column here, column G, and checks whether or not there's a 0 there. It says if it's not 0, then cell, then it takes, if it's not 0, and if B25 is equal to a Q, then this one's broken, sorry. Let's use this one. Yeah, this one looks here at G27, which is here, it's a 1. If it's not 0, then it asks if B27 is equal to A11. B27 is an A. A11 is an A. So they are the same. So it comes up with a true condition, and then it puts the formula in, which is B11. B11's right here. So it basically put this formula in that position. So to actually make this work for all 21 residues, this if statement would need to be longer. So it'd have to cover every possible residue. So instead of just saying A11, we would need like A12, A13, A14, and B12, B13, B14, and so forth, until all the formulas are covered, and then repeat for the other. And that would give us a list of all the residues that differ. And then count the total differences. That's basically just summing up column G. It's adding up all the zeros, which are falses, and the ones, and counting them, and they're totaled over here. Okay. That's pretty much it. This might seem kind of involved, but really if you just play around with it using like half a dozen or so residues and get everything to work the way you want, it'll go a whole lot easier than pasting 1200 in there to start with. Okay, are there any questions?
Mic on, mic off, mic on.
Do you want us to do that from a blank like Excel document and just set the whole thing up ourselves, or is there a template that we should go off of?
Oh, I'll post the one I just showed you.
Oh, okay.
You can start from that.
Mic off.
And I should mention that, see, this sort of information, like looking at two sequences, is part of drug discovery. And chemistry, especially chemistry, where we look at those exact interactions between proteins, is actually done in pharmaceutical companies. And there's ways to do that computationally, and to do it pretty accurately. And that way experiments can be done, so to speak, without being exposed to anything dangerous, like if it's a virus or if it's radioactive and things like that. So it's becoming more and more prevalent in drug discovery. So when this is all said and done, then we'd have COVID-19 spike protein compared to some other one. Let's say it's a bovine, or it could be a bat spike protein. And then have a list here of all the differences, and then it will show the chemical structure in each area that's different. And then those could be lined up with ACE2, which is the problem that, COVID-19 spike protein binds with ACE2. Can line ACE2 up, not in this project, of course, but that could be done, and then find chemicals that could interfere with that could like disrupt that binding and keep COVID-19 from doing what it does. Does anyone not have Excel? If not, you'll need to let me know. So everyone should have it through.
I think we automatically get it with our email.
Okay, I thought so too. And it's handier to have a computer. I don't know how many of you are operating off a phone, but if you don't have access to a computer, be sure and let me know.
Dr. Clark, I lost track of time and logged in a little late. I was wondering, are you going to add, along with the template, like some instructional guidelines, or is this our only source of instruction on how to do it?
Yeah, I'll post some guidelines also. Yeah, I'll have it up there Wednesday, by the end of Wednesday.
And when is this due? I apologize. You probably already explained that.
Yeah, it's May 8th.
Thank you.
Okay. Okay, are there any other questions? Oh, and as far as the COVID-19 spike protein sequence, there is one available. I'll just need to convert it over so it could be used for this project. And so I'll have that up probably Wednesday also. Okay, let's see where we were here talking about the class project. So let's look at the rest of chapter 16. There were only a couple of examples relating to Gibbs free energy that were left. Okay, this was example 18, where we have a reaction involving calcium carbonate. It's decomposing into lime, which is calcium oxide, and CO2. This is a reaction that occurs in a lime kiln, by the way, where they take limestone, like in the bluffs in this area, and they bake it in a kiln. If you heat it up hot enough, it'll get rid of the CO2 part of it and make lime. So then the lime is packaged up for use in cement. So in this problem, we're asked to calculate the equilibrium pressure of CO2 at 298 K and 800 K. So first, let's look up the thermodynamic properties for each of these compounds. That's what this little table is. It's pasted from the table that's in the back of the class notes. So this has delta H, delta G, and standard entropy for each of the three. So for part A, let's first calculate delta G. And so delta G is the Gibbs free energy of the products, which is the calcium oxide and CO2, which is minus 604.17 and then minus 394.4. So we add those up and subtract what the reactant value is, and we end up with 130 kilojoules per mole. And then now that we know delta G, we can get the equilibrium constant through using log Kp as minus delta G on RT. So we plug all that in and we get minus 52.6. Now, let's find what Kp is. To get Kp, we would do the inverse of the log operation to both sides. In other words, take the exponential of both sides. So that says Kp is equal to e to the minus 52.6 power, which is 1.4 times 10 to the minus 23. Now we need to know how Kp relates to the pressure of CO2, because the question specifically asks for the pressure of CO2. So let's write the K reaction for this, or K expression for this reaction. And if we did that, we'd see that only the gas remains, everything else is a solid, so it doesn't appear in the K expression. So CO2, the partial pressure of CO2 appears in the numerator, and that's all there is to K. So K and PCO2 are equal. So the pressure of CO2 is 1.4 times 10 to the minus 23. And that's a little different from the answer that's given here, but I'll explain why in a moment. I think there's a typo in one of the answers in the textbook I got this from. So let's look at part B. Okay, part B is repeating at 800 K. So basically we set up the identical expression, but instead of 298, we have 800 Kelvin. And then solve for log Kp and we get minus 19.6. So there's a pretty big difference between the two temps. And then solve for Kp and we get Kp is 3.2 times 10 to the minus 9. So in this case, the pressure is 3.2 times 10 to the minus 9. That differs from the answer given in this book also by a little bit. So to find out why, I double checked the calculation that was given in this book and found out there's a difference. I think this, where it says 176.1 for delta H, it should be 178.1. So I think that 6 is a typo. So on your notes, you might want to scratch through the 6 and write an 8 above it. Okay, if no questions, I'll proceed to the next one. The next one is a little more involved than this one. Okay, the next one is example 19. So we have a solid and the molecular weight is given. It's dissolved in water, which cools the water. And we're given the solubility max. And from all of that, we're supposed to find the standard entropy change. And this is very similar to experiment 19. So we have a strategy to tackle this. It's 4 steps. Okay, first we would calculate the delta H from the heat loss by the water and the number of moles of solid. Then we calculate K, that's the reaction quotient, from the saturation point. Then we get delta G from K, similar to the previous example. And then now that we have delta H, delta G, and the temp, we can get delta S. So the last step would involve putting all of that together to get delta S. So for step 1, calculate delta H. So delta H is equal to the heat added at constant pressure. And the heat that's added, we can find out from the temp change of the water. We're given the mass of water, which is 78 or 75 grams, and that's temp change, which is 12.9 and 25.4 for the range. So we use the standard mass times the specific heat times delta T. Mass is 75 grams. The specific heat is in tables. We're using joules. So it's 4.184 joules per gram degree C. And then multiply that by the temp change, where the final temp is always listed first. So it's 12.9. It's a final temp minus the initial temp, which is 25.4. So we end up with a negative number here. It's minus 3,920 joules. The negative sign means that heat was emitted by the water. And that is equal to the amount of heat which is absorbed by the reaction. It's like the reaction basically sucked heat out of the water. So the reaction would be endothermic. So now we can calculate delta H. We know that it's a constant pressure because they talk about standard conditions and they don't mention pressure at all. So we can assume it's standard pressure, which is 1 atmosphere. So the delta H is equal to minus Q, which is plus 3,920 joules. And we reverse the sign because it's opposite to what the water did. For the water, heat was lost, but for the reaction, it was gained. So we have a plus sign for delta H. But for delta H, we need to know how many joules per mole. So we need to find the number of moles. So the number of moles is the mass of the sample divided by the molecular weight, which is 71.242 grams, which is 0.0387 moles. So we divide all that and we get 101 kilojoules per mole. So that's what delta H is, 101 kilojoules per mole. Okay, another page here, second step, calculate K. So here's the reaction. We have some unknown solid, we'll call it X, in equilibrium with its aqueous form. And we know that equilibrium can only occur when it's saturated, right? We can't just put a little bit in there and calculate a K because there has to be solid present. So we have to basically keep putting X into this solution until it won't dissolve anymore. And at that point, we'd have solid in equilibrium with the aqueous state. So K is equal to the reaction quotient, which is X, the aqueous phase, and we leave out the solid phase. So this concentration we can get from the maximum solubility, which is 954 grams per liter. And we convert that to moles using the given molecular weight. And we end up with 13.4 moles per liter. Okay, now step 3, we can calculate delta G from K. So delta G is minus RT log K. If we plug all of that in, we have R, the gas constant. The decimal points moved over 3 spots, and we use kilojoules instead of joules. So that's 0.008314 kilojoules per mole Kelvin times the temp, which is standard temp 298 K, and the log of K is 13.4, and all of that comes out to minus 6.43 kilojoules per mole. Now we can finally calculate delta S. So delta S is equal to delta H minus delta G on T. And if we plug in everything from the previous steps, we have 101 minus the minus 6.43 divided by the 298 Kelvin. And that comes out to 0.359 kilojoules per mole Kelvin. Or if we wanted to express it in joules, it would be 359 joules per mole Kelvin. And that's close enough to the given answer. It differs slightly because of rounding. So that would be the entropy associated with dissolution of the solid. So it's positive because there's much more disorder associated with a dissolved solid than a solid form. That's all I had for chapter 16. Were there any questions? Remember the quiz on Wednesday covers not what we just did, but everything up till then. Everything we covered through Wednesday. So if you have a question on any of that, now would be a good time to bring it up.
Mic on.
So pretty much just the beginning of chapter 16 is what's on the quiz.
The beginning, it's a little more than just the beginning. Let's see, it's listed here.
Okay, but it is just all chapter 16.
Yeah, pretty much. It's through page 14 of the class notes.
Okay.
And that is through example 17. There are 3 examples, 18, 19, and 20, which won't be on it. They'd be on the exam instead.
Okay.
Mic off.
Okay, well, let's go on to chapter 17 then. Okay, this is just the initial part of chapter 17, and it's based on the class notes that were put in Blackboard. And by the way, we'll be using Jesperson mostly instead of OpenStax. And Jesperson's available on Wiley Plus, and it's chapter 19 in that textbook. And they do a really good job on electrochemistry. See, this. Yeah, this is the class notes which are on Blackboard. So this part goes over the basics of redox reactions and how to balance redox reactions, which I assume most of you have already done in your prior chemistry class. But we'll just go over a little bit of it as a review. Okay, electrochemistry involves the conversion of electrical energy into chemical energy. And a really good example of that is a battery. So we charge a battery up with electrical energy, and that converts it to chemical energy. And in return, then it releases the chemical energy in the form of electrical energy to power a laptop or a phone or whatever. Okay, there are some definitions to keep track of. Firstly, we have oxidation. Oxidation means losing electrons. For example, potassium, when it's dissolved, becomes K plus and loses its electron. So it loses the electron and therefore is oxidized. And the term for gaining electrons is called reduction. An example of that would be bromine acquiring 2 electrons and becoming 2 bromide ions. The oxidizing agent is what's reduced. And for example, if lithium and fluorine reacted to make lithium fluoride, the substance that's reduced would be the fluorine, because the fluorine ends up with an electron, lithium's electron, it takes it away. And the oxidizing substance would be the lithium because it had its electron snatched away from it. And redox is the slang term that refers to reactions involving oxidation and reduction. Where we have oxidation, we have to have reduction because the charges have to balance. If, for instance, this reaction right here, we have on the left side, we have an overall charge of -2. So on the right-hand side, we have to have an overall charge of -2. In the top reaction here, we have a left side, which is neutral. And on the right side, it has to sum to 0 to match the left. So we have +1 and -1, which sums to 0. So anytime we have a loss of an electron, something else has to gain it. So reduction and oxidation always occur as a pair. And another term is oxidation state, and that refers to the charge that an atom would have if both electrons in each bond were assigned to the more electronegative element. And there are rules for assigning the most electronegative element, but they're pretty much, they can be predicted by where atoms or elements live in the periodic table. Like here we have a periodic table, and over here on the right-hand side, that's right next to the noble gases. These are the halogens, fluorine, chlorine, bromine, or bromine and iodine. Those are really electronegative, especially fluorine. You know, they're so close to the noble gases. All they need is one more electron to reach that state, which is stable in an atom world. It's like maybe it's like people who make enough money to do whatever it is they want to do. Like if they wanted a house or something, so they try to make enough money to get that house. So this in Adam's world, they try to get here. Fluorine wants to get over one more spot by gaining an electron. So it's at the top right corner. It's the most electronegative element we have. It's measured on the Pauling scale as 4.0. So in the right upper corner, the periodic table, those are the elements that are really good at taking electrons from other elements. So these are going to be negative. So these elements will be reduced. They'll gain electrons. And the ones they most likely take it from live over here on the other side of the periodic table, like lithium, sodium, potassium, and the alkali metals. They all have a small electronegativity. It's like 1 or less. And there, if we wrap the periodic table around like a helix, like it is some in some versions of the periodic table, it's a helix, which it really is. Lithium would be next to a noble gas, and it could lose an electron to become stable. Just like fluorine gains an electron to become stable, an alkali metal can lose an electron to become stable. So they like to lose electrons. And over here, these elements like to gain electrons. So together, they work well together. So lithium loses an electron, and it becomes oxidized. And at the same time, if it attached to fluorine, fluorine would gain an electron and become reduced. So we can write chemical reactions and balance them, and just simply in terms of what's oxidized and reduced. And for example, here's a reaction involving phosphine. It's combusting phosphine, yielding P2O5 in water. So we can look at a reaction and identify which elements are oxidized and reduced. So if we have a look here on the left, we have phosphorus attached to 3 hydrogens. Hydrogen is assigned a +1 oxidation state. So if there are 3 hydrogens and each one of them is +1, that would make phosphorus a -3. So phosphorus is a -3 on the left side of the reaction. And if we go on the right-hand side and look at phosphorus, we have 2 phosphoruses attached to 5 oxygens. An oxygen is assigned a -2 because here's oxygen and it's 2 columns away from the noble gases. So oxygen's -2. Over here, hydrogen's 1 column away from the noble gases, and we give it a +1 charge. So in P2O5, we have 5 oxygens times -2 gives us a -10 charge for oxygen total. And that's distributed over 2 phosphorus atoms. So we have -10 divided by 2, which would be -5. That's a charge that the phosphorus has to balance. So each phosphorus has to be +5. So on the left, we have -3, and on the right, we have +5. So if phosphorus goes from -3 to +5, it lost electrons. So here, phosphorus is oxidized. And if we went through and looked at oxygen, here O2 is in its standard state. We would assign that an oxidation number of 0. And over here, it's -2 in P2O5, and it's also -2 in water. So it goes from 0 to -2, which is a gain of 2 electrons. Therefore, oxygen is reduced. So this sort of reasoning is the basis for balancing reactions based on the oxidation number change method. And I won't go work examples since presumably it popped up in your prior chemistry class, but it shows step by step how to balance reactions by that method. Now there's another method which involves splitting reactions into 2 half reactions, and it's called the Ion Electron Method. This one's used more so than the other, or the oxidation number change method, and it involves about 6 steps where we operate on each half reaction and balance them, and then at the end combine them to get the balanced reaction. And we can do that in an acidic solution. And in that case, we balance with H pluses since we're in an acid. So we insert H plus as needed until we get it to balance. There's some example problems you can try for practice. And then we can also balance them in basic solution, and it involves using OH minuses. like in this step right here, we use OHs. We start out with H+, but then towards the end, we add an equal number of OH minuses. And we end up with a balanced reaction and basic solution. And the one I like the most is by far the most powerful, and I think also the easiest. But the problem with it is that it doesn't really explain the chemistry. Like the Ion Electron Method and acidic and basic solution involves half reactions, and it shows what's really going on. But this Algebraic Method is really powerful, but it doesn't like shed light on the underlying mechanisms. So this involves treating a reaction balancing problem more like math. So for example, if we had this reaction, which is unbalanced, and we needed to balance it, we would put variables in front of each substance, A, B, C, D, E, and F in this example. And this is the same example that was used at the top of this document. The oxidation number change method had an example, same example as this one. So we can balance that same reaction using the algebraic method. So we have these variables, and we make a table and count the number of elements left and right. So we, for instance, here, hydrogen, we look at the left side of this reaction and count the hydrogens. Well, we have A hydrogens there. So on the left side, we have A, and on the right, hydrogen shows up here in the waters. So we have 2F hydrogens, 2 times F. For example, if F was 3, then we'd have 6 altogether, 2 times 3. So we have 2F hydrogens on the right. And then for chlorine, on the left, we have A again, and on the right, we have C from here. And then in the second term, we have 3D. In the third term, we have 2E. So on the right, we have a total of C plus 3D plus 2E chlorines. And if we continue on and fill out the rest of the table, we end up with it looking like it's shown here. So to balance the reaction, we set the left side equal to the right side, like in this table. And now it looks just like math. Really, we have 6 unknowns. That's A, B, C, D, E, F. That's 6 unknown variables. And we have 5 equations. So we have 1 extra variable, which means that we can guess one of them and then solve for the rest. So let's choose a convenient one to guess. Let's have a look down the list here and notice if we choose F equals 1, that would make A equal 2. And it would make, if F is 1, then B would be a 7th. So it makes things simpler. We could choose any number we wanted, but it's easiest to choose the most convenient. Like we could choose F as our guess. For example, we could guess D equals minus 0.003818. And we could work through all the math and get the exact same answer, but have to work a whole lot harder. So let's choose F equals 1. So if we choose F equals 1 here in this part, right here, if F is 1, reaction 1 says that A must be 2. And reaction 5 says that B must be 1/7. Okay, and if B is 1/7, reaction 4, we plug in 1/7 for B. Reaction 4 says that D is 2/7. And reaction 3 says C is 2/7. So now we have values for A, C, D, and F, and B. So we have 5 of the 6 already. And now if we use the second reaction, plug everything in, we get E is 3/7. So then write all the coefficients in order, A, B, C, D, E, F, clear the fractions by multiplying through by 7, and we get coefficients of 14, 1, 2, 2, 3, 7. And that matches the first example. Could have a look up here. Yeah, this is it here, 14, 1, 2, 2, 3, 7. So we get exactly the same answer as before, only I think it's a lot easier to get the answer. So there are a couple of examples here you can try for practice. Okay, any questions? Okay, so let's go to the Wiley plus Jesperson. Okay, this is the top, section 19.1. It's about galvanic, or AKA voltaic cells. And those have become what we call batteries. And the energy from a battery comes from a spontaneous redox reaction in which the electron transfer is forced to take place through a wire. And let's see if we go back up here to this table of electronegativities. And if we wanted to build a battery out of some elements, we would pick an element where there's a significant difference between their electronegativities, because in that way it would most likely be spontaneous. For example, one electrode of a battery containing oxygen could be used in combination with an electrode containing lithium. That we'd have a difference of 3.5 minus 1.0, just a difference of 2.5, which is pretty significant. And that's essentially what's going on in your lithium ion batteries and your computers and cell phones. Because one electrode that is carbon-based and the other is oxygen-based, and in between them, lithium is shuttled back and forth. Okay, so in this example here, it shows that effect. In your first chemistry class, this was called single replacement. And one way to demonstrate that is to put a copper wire inside a solution of dissolved silver, for example, silver nitrate. So that's put in solution, and then a copper wire is immersed in it, and immediately the silver starts plating out on the copper. And it ends up looking like this figure on the right, where we have this silver caked all over the wire. And the redox reaction is written as follows here. It's 2 silver ions combining with copper solid yields copper ion plus solid silver. And in other words, the silver and the copper traded places. The copper was in the wire to start with, and afterwards it's in solution. Initially, the silver was in solution, and afterwards it's on the wire as a solid. So that's why in the first chem class, they called this single replacement because it's really just the two metals trading places. And the reason they would do that is because if we look here in the periodic table where copper, silver, and gold live, they happen to be in the D9 column, the transition metals, and those are known as noble metals. They're all extremely stable, especially gold. Gold won't tarnish. I mean, it's been used on roofs since the Middle Ages, and they don't tarnish. It's just extremely stable. And silver is not as stable, but it's still more stable than copper. So if silver and copper compete for electrons, like they would in that solution in the beaker, silver would win. Silver needs the electrons more. It can be much more aggressive and achieving its stability than copper. And if we put gold and silver together, the gold would win. So in the beaker that had silver and copper, silver won, copper lost. So copper loses its electron and goes out into solution, and silver gains its electron and becomes a solid again. So whenever the reaction proceeds, the solution turns blue in color. It's a characteristic of copper solutions. So this reaction can be divided into two half cells. One half cell we have silver, and in the other half cell we have copper. And in silver's half cell we have reduction because silver gets its electron back and becomes solid again. And in the other cell, the copper becomes oxidized and loses its electrons. So a battery takes advantage of this situation. We can have half cells, keep the two solutions separate and connect them with a wire and a switch. So then the electrons have to travel through the wire, and then we can power a circuit through that. Here's an example where we have the silver solution on the left and the copper on the right. They're both connected. You have a cathode, an anode. The anode is the negative electrode. Cathode's the positive electrode. Reduction always occurs at the cathode, whereas oxidation occurs at the anode. So the two electrodes are connected to a circuit with a switch. Like on your phones or your laptop, this would be the power button. So whenever the power button is switched off, it's like throwing this lever up. It interrupts the circuit. When you push the power button down, like when you turn it on, it's like closing this switch, and it allows current to flow through the wire. So we can combine the two half cell reactions as shown here. Silver gets reduced. So each silver gains one electron, becomes solid. And the oxidation half cell has copper being oxidized. Each copper atom loses 2 electrons. So to keep the total number of electrons the same, on the left side versus right, we'd multiply the top one through by 2. So once the 2 is in place on top and bottom, we can add them, and then the 2 electrons cancel out left and right, and we're left with a balanced redox reaction, which is called the overall cell reaction. There are a couple of parts in this picture here. Should be mentioned, the salt bridge is usually a gel, kind of like in biology, augers used a lot. And in batteries like cell phones, there's a thin strip that's called a separator. It's a really thin, tough sheet of plastic, and it allows ions to travel through, but it doesn't allow one electrode to touch the other. That's why it's called a separator. But when we use beakers, it's called a salt bridge. So the salt bridge allows ions to travel through without the solution actually leaking from one side to the other. So whenever copper is oxidized, so the electrons travel up the anode and they come to this wire. They can travel through the wire because it's usually a metal. Electrons can easily travel through metals, and then it travels down over here on the cathode side, and it becomes available for the silver. So silver sees these electrons accumulating and goes over and grabs one. In fact, it's sort of like a tug of war. If we envision like somebody on the left named silver and someone on the right named copper, and which one needs electrons more? The one in this case would be silver. So silver is extremely stable in solid form compared to copper. So silver is going to be the stronger one. It will take electrons out of this region here, the cathode. It takes an electron out of it. It works its way through the wire, and it forces the copper to oxidize here and to replace that empty spot where there was an electron. So copper gives it up, and that process continues over and over again, and we get a current going from left to right. So here's a zoomed in version of what's going on at the cathode and anode. The nitrate here is a spectator ion. It doesn't do anything except hang out. And electrons accumulate on the cathode. That's where the silver ends up getting its electron from. So silver, which is positive, is attracted to the cathode. And then the anode was over on the copper side. The anode is where the copper becomes positive. So we have a standard notation, use 4 cells like this. It's consists of 4 parts. It's the 2 half cells. So in this case, the copper's on the left, silver's on the right. So we have anode and then a space, like these 2 hash marks stand for the salt bridge or the separator. And on the right, we have the other side. So actually anode on the left, cathode on the right. So we have copper, the copper electrode, solid, is the actual anode. And it's in copper 2+ solution, immersed in it. So in other words, copper solid in contact with copper 2+ solution, separator. Then on the other side, cathode side, we have silver solution in contact with a silver electrode. So that's the standard notation used to describe cells. And here it's described in more kind of like what I just said, but it has all the definitions shown. If we had a cell consisting of iron and zinc, it would be written with zinc solid in contact with zinc solution, separated. And we'd have iron 2, iron 3 in aqueous solution, contact with a platinum electrode. So in this case, we could carry out a reaction between zinc and iron using a platinum electrode, because the electrode actually can be made of anything that doesn't interfere. In this case, it doesn't have to be made of iron. Platinum's really stable. In fact, it's more stable than gold, and it won't react. So it's handy to use as an electrode for that reason. All it has to do is conduct electrons. See, there's more about half reactions here. The book does a really good job, I think, of explaining this. So be sure and have a look at it. So it goes through more examples. Here's an aluminum nickel cell. Then it leads up to 19.2, where we start talking about voltage, cell potential, current, amperes, milliamps, and cell capacities, and all of that stuff. So that's where we'll pick up on Wednesday. It's 19.2. Okay, any questions?
Mic on.
I don't have
Are
any.
you all asleep? Hopefully not.
Yeah, not quite, not yet.
Okay, well, that's a positive sign. Okay, well, if no questions, we can stop for tonight and pick up next time.
All right.